Massimo?
Thank you for the nice introduction. I would like to also thank, I don't know whether Ari or who else, for the title, Preclinical Therapeutics with Novel Mechanisms of Action. Okay, it's everything and nothing. And so what will I do? Disclosures. Okay, let's start with the landscape of what is going on in terms of targets for treatment. And yes, Great Expectations is the original one, the 1946 film. We have a lot of excitation, but up to now, truly, nothing really ended up as we hoped. And let's go a little bit more deep and think about the biological correlates of cure. Essentially, we have, I will not enter into the definitions of partial, functional, complete, sterilizing cure. Mechanistically, we have three main mechanisms: silencing cccDNA, reduction of the pool of the cccDNA, elimination of infected cells. The questions are what we want to do, control or cure? What to target, what to prefer? Obviously, we go for functional cure because we are not able today to think about something more. And then, is cccDNA silencing possible? Is cccDNA elimination possible? And is silencing propedeutic to elimination or the other way around? Going from the clinical point of view, we can classify the whole thing in a different way. We have a lot of drugs that do different things. We can think of them as inhibiting replication, diminishing antigen load, or stimulate the immune system. I will not further discuss the first two categories because somehow, especially the first one, because other people will do, and there is a lot of clinical work on that. So I will try to stay away from the clinical data and try to reason on other points, about other points. Just I want to say that blocking replication, cytoplasmic replication, capsid-related replication is important, and it may affect indirectly cccDNA de novo synthesis, the intracellular recycling, and the important coating of relaxed circular DNA containing capsids. And this is important, and entry inhibitors and CAMs may inhibit cccDNA establishment. I will not go into that because it was already covered. For the antigen reduction, you just heard Jordan, it may restore innate and HBV-specific adaptive immune responses. We have few data that prove this, and we will hear this afternoon about the data that were just mentioned by Jordan with the Arbutus drug, that are some of the few data that we have directly from patients. So I will focus essentially on three things, and you will see how I will do it, and probably will smile later on. So targeting cccDNA, targeting HBX, and immunotherapy, immunomodulation. Why I added targeting HBX? Because X is a crucial protein in the control of cccDNA function, and this comes from a lot of studies. And if we go a little bit deeper, I will touch first the HBX inhibition with different approaches, its relation with silencing of cccDNA transcription, and later on I will go to strategies that go directly on cccDNA stability. First question I want to a little bit to address is, can cccDNA be silenced or epigenetically silenced? What we know from clinical studies is that transition from to the E-negative chronic hepatitis infection is associated with the reduced cccDNA transcription activity. And we define cccDNA transcription activity as the ratio between the number of CCC molecules and the amount of 3.5 transcripts, because this is what really defines whether or not there is transcription from the cccDNA. And this was showed also in several other papers, including a paper that probably Barbara Testoni will mention this afternoon because it's related to biomarkers. But there are data that confirm that with E-negative status, cccDNA transcription activity goes down, but does not go completely down. And there are data that in patients with long-term NUC treatment, again is a paper from Barbara Testoni's group, you have indeed a silencing of the cccDNA with epigenetic changes. And the two arrows there in telbibudin-treated patients is related to cccDNA chip, in which some histone marks of repression, K27 methylation, is up, and the corresponding K27 acetylation is going down. So there is some epigenetic control. And the last panel are some data, recent data that we generated from patients analyzing the liver biopsy from patients with a functional cure versus chronic hepatitis B E negative. And as you can see, there is much less cccDNA, much less DNA in the liver, in the bulk analysis of the liver samples. You have less 3.5, but if you look at the transcriptional activity, in functional cure patients, you still have some patients that have fairly high transcriptional activity. This means very little cccDNA molecules there, but they are active, and this is something that we have to keep in mind. And so if we go to targeting HBX, because it's the crucial protein that regulates the activity of the cccDNA by inducing the degradation of the SMAC5/6 restriction factor. If you target the interaction between HBX and DDB1 degradation complex, you indeed have a less transcription, but the effect, as you can see, is not dramatic. And this was obtained with a drug that came from a screening, but it's something that the people working on hepatitis C know very well, and did, because it was used as an anti-limiting that was used in hepatitis C before the real direct antivirals arrived. And you can also target the NAD8 as another pathway. I have no time to go into all the details. The inhibition of NAD8 with a specific drug lowers cccDNA transcription and lead to cccDNA destabilization. Look at the effect. Is 30%, 40% is not big. And targeting the interaction between HBX and the 20S proteasome, again, and this study was published in Hopital, and there's also a part of the work done in vivo in mice, dicumarol and NQ01 inhibitor blocks cccDNA transcription and promotes, by promoting HBX degradation. So there are indications that you can work out a block of HBX. This is completely preclinical. We have no idea how to convert this in something that may work in patient yet. And some interesting data come from the work of Maura Dandri's group. They published this year in Gut a paper in which they showed that by using siRNAs, the same siRNAs that we use in patients that you heard about, since the target of this si actually go to a region that induce the degradation of all transcripts, they demonstrated that in these chimeric mice, indeed you have less transcript for X protein, and you have less X protein, and by doing this, you can induce again the activation of the cccDNA. So it's the proof of a concept that siRNA may have a second mode of action in which, besides HBV RNA degradation, you can hope that there is some cccDNA transcriptional silencing. But if it is true, why this does not happen in patients? Because finally we have no idea. Or we didn't look well, or there is something else that is not really clear for us. And here, again, I was scooped by before, because somebody already said that interferon is coming back. So I used the DeLorean 1985 car, renamed PEG interferon, and Back into the Future. Interferon is coming back. It's coming back in the delta field. NAPs work, but they work because interferon is there, I suppose, or I fear. But we know from the Stefan Urban work that interferon has a very special activity in delta infection because it's able to block NTCP-independent HDV spread. And this is why bulevirtide plus interferon works better, whether we like it or not, but it works better. If you look at the graph from the NAP studies, you have in the red rectangle a phase of the treatment in which interferon is not yet there. the good or bad ALT flares are not there. I agree with Jordan that talking about ALT flares is not biologically relevant. It's clinically perhaps important, but biologically it does not help us to understand. And in that phase, HDV RNA is going down. So perhaps NAPs have some other activity besides entry inhibition or post-entry inhibition, as was demonstrated in some models. That has nothing to do with HbS decrease and ALT flares that induce the functional cure, and maybe there is other activities directly on the replication cycle. Let's go back to the animals, to the chimeric mice that were studied and I presented before from Maura Dandri. They showed that the PEG interferon is indeed doing the same thing that siRNA. There is less pegylated interferon in this model, and PEG interferon is able to induce again to target SMAC5/6 and to induce the re-expression of and the decrease, the block of the HBV RNA production from the cccDNA. And this was just a confirmation of a lot of data that were published before. We published that before, besides the well-known activities on the stabilization of RNA-containing capsids, besides the activity that is there but is slight and not very big on cccDNA stability through deamination. There is a clear epigenetic effect on cccDNA with silencing. Whether this is in all patients, we don't know. Whether this is contributing to some of what we were seeing in patients, perhaps yes. There are a lot of other drugs that are controlling the epigenetic of the cccDNA, but forget about them, because they're not specific. There is a lot of problems, complexity of responses, functional redundancy, duration of response, off-target effects. So we will not go there for silencing. But looking really at the preclinical data, we are coming to a scenario that looks more complicated than what we were thinking before. Because in a study that was presented at the JGB meeting 2 years ago, Mauro Dandi's group showed that shutdown of HBV transcripts promotes the appearance of SMAC5/6. I already told you that, and cccDNA silencing, that's okay, but there is no change of the post-translational modification on the cccDNA, of the histones on the cccDNA. So there is no active repression, but more something like cccDNA compaction without active repression. This was what they proposed according to what they were seeing. And there is another paper that was published last year from Zheng Yuan group in Shanghai, in which they showed that with interferon, the accessibility of cccDNA to MAC-bind nuclease is much reduced. So again, you have something that cccDNA is less accessible after pgRNA, pegylated interferon, but there is no active repression probably. And what does it mean? Let's go for a cartoon trying to explain that. On the left, you have an active cccDNA with the transcription factor, coactivators, and acetylation marks. On the left, all the possible flavors of silencing. You may have a cccDNA that is open, but is deserted because there are no transcription factor, no coactivators. So the acetylation marks can be there or not, is not important. And you have the other possibility. I didn't see before the clock. Sorry. The other possibility that you have a compacted cccDNA. What was proposed by Mauro Dandi's group with siRNA, loss of X, but no changes of the cccDNA epigenetic control. And you may have a repressed, like in the long-term NUCs in the paper from Barbara Testoni group, in which you have some post-translational modification of the histones. And all this may have a lot of impact on what we are doing, because probably we are treating as silenced things that are different, and we are trying to change and to use drugs to do things on a completely different scenario of population of people. And it's like in the clinical trials. If you put everybody in, you may lose the effect, and if you do you select too much, you only treat 1% of the population. But we have to know before to make the decision. And in HIV, as was already mentioned, people are using the so-called the shock and kill. Perhaps we have to try to do something similar in HBV, like shock and degrade. Try to open the cccDNA or make it accessible. And indeed, this has been done in the same paper I mentioned before, by Zheng Gongyuan in Shanghai. They used the histone deacetylase inhibitors, and they obtained some more accessibility and degradation of the cccDNA. I wanted to tell you a few things on the direct targeting of the cccDNA, but I was too long. So I just will go to one single slide. And it was already mentioned, the potential for direct targeting, genome editing of the cccDNA, a lot of limitation, a lot of strategies to change this, and possibly one new approach may be base editors as compared to the classical CRISPR-Cas9 approaches that are catalytically disabled or inactivated Cas9 that generate single-strand breaks and not double-strand breaks, coupled with the deaminase that is able to act on single-strand DNA to edit the genome at the specific target site. And at the HBV meeting a few weeks ago, the BEAM team, in collaboration with Fabian Zulim and Barbara Testoni, they showed indeed that you can inactivate cccDNA in vitro and in vivo, introducing specific mutation that abrogate HBV replication and silence viral protein expression. The big point here, like for the other approaches, is what is the fate of the cccDNA. Barbara Testoni Guada Martinez published a paper in which they showed that the residual cccDNA that is strange, is manipulated, is edited, is still transcriptionally active. So we have really a problem there. Immunotherapy, I will not touch it because I was afraid to have I didn't dare to put Georg Lauer photo there, but I could do that with Adam, so I will not talk about that. And finally, the only slide before ending was this one, and it's already been mentioned. The epigenetic scar is very important. We have really to go into that. And there are also few evidences that something similar is also going on in hepatitis B in addition to hepatitis C. Thank you very much.